Thank you for your very kind introduction, and thanks to Aaron, Christina, and the rest of the organizers for the honor of allowing me to present to you today. And I'm going to start with a historical perspective on where we have come as a field. And so at Genentech, we started in therapeutic antibodies in about 1990, and in the sort of lexicon of the previous talk, this was really in the skepticism phase. You know, we'd sort of looked at monoclonal antibodies in the clinic, and most of them almost invariably had been immunogenic and really hadn't worked very well. And so then there was a period in the 80s with the advent of chimerization by Sherry Morris and humanization by Greg Winter and also Kerry Queen at PDL. And then there was this sort of phase where, while we were trying these new tools, we didn't actually know whether they were actually going to be useful or not. And so that kind of reflects this real sort of lag phase in the field. And then in the late 1990s, this is where things really started to take off in terms of approval of these antibodies. And we sort of reached the astonishing total of 54 approvals, either in the US or Europe, and 49 of these are still marketed. If you look closely, there was kind of an inflection point last year. There was actually a bolus. There were 9 antibodies which were approved, and there actually already have been 3 this year to date. And there are roughly 400 antibodies in clinical development. So most of these antibodies have been improved in one of 2 settings, either in oncology or autoimmunity and chronic inflammatory diseases. And there are also antibodies starting to impact several other areas. From a commercial perspective, the worldwide revenues somewhat lagged behind the approvals and really started to take off about 2000, and then reached a sort of a linear growth for the last few years, and reached $75 billion in 2015, and the projection for the next few years has actually been even faster growth. And actually, this is projected based on some of what we heard in the previous talk around antibodies in cancer immunotherapy. If you look at the world's best-selling drugs, 5 of them are antibodies. They're shown in orange in this slide, and actually there are 2 other biologics here. And these 5 top-selling antibodies on their own contributed $40 billion in worldwide sales last year, which is actually more than half the antibodies' total sales. So a deceptively simple question is, well, how do antibodies actually work? And a few years ago with Andy Chan in a review, we tried to cover what was known at that time. And there are a few of the mechanisms that were sort of fairly well established. So blockade of receptor or ligand, downregulation of receptors, depletion by a variety of different secondary immune functions, signaling. And one which we didn't highlight, and frankly I should have done it, is around induction of adaptive immunity. And actually this was something that was really called out by Greg Adams and Lou Weiner in a really seminal review in Nature Biotech in 2005. One way of exploring the sort of mechanism of action of antibodies is to look at the prescribing information. And so if you look at the prescribing information for all of the antibodies that are improved in autoimmunity and inflammatory diseases, what you'll find is that most of them block receptor or ligand. There are a few which also deplete target cells. In oncology, the situation is a little bit more complicated. And consider first there are antibodies which directly target tumor cells, and the majority of cases here, one of the mechanisms of actions is by direct depletion of cells. But there are also other mechanisms of action too. So, for example, there are several antibodies which are approved for EGF receptor overexpressing tumors, and as well as depleting cells. Several of them also block ligand interaction with receptor, downregulate receptor, and in some cases they have other mechanisms of action too. Down the bottom are some of the antibodies that we heard about in the previous presentation, so immune sort of modulators, and we've also got Avastin, sort of an antigenic, and another anti-angiogenic. And even this table really is kind of a gross simplification. It's actually kind of very difficult to understand sort of mechanism of action. You can measure antibody sort of properties. You can do that in vitro. You can also do it in vivo. That's not the same thing as saying that you actually understand how they work in patients. So let's consider antibodies from the perspective of what they do well and what they don't do so well. On the strength side, they're often very easily generated to targets of interest. They have many properties which are readily tunable, and so this in general is a good thing because you can really tailor their properties for particular applications. Although in a way it's sort of a double-edged sword because sometimes you don't actually know what the most desirable property is. They're typically well tolerated. The success rates, well, that depends how you actually measure it. There's so many reviews in this area from Janice Reichert and from a few years back. This is actually not from Janice, but from another group, but it's a little more recent data. So the approval rate is roughly 14% from phase 1 to approval, which may not sound very high, but is typically higher than for small molecule drugs, which is typically in the high single digits. Antibodies. If one looks at the antibodies which are approved, almost all of them are IgGs. But in a way, this is quite misleading because it's kind of looking in a rear view mirror as to where we have been as a field. I'm struck in coming to meetings such as this, really just the diversity of different platforms which exist now and also are continuing to be sort of developed. So I hazard to guess if this talk was given in another 10 years' time, that this actually would be quite a different picture with a much broader portfolio of different platforms that have made it all the way through from this kind of the technology development through clinical development to approval. Now, of course, antibodies also have their limitations, and these include high cost, inaccessibility to intracellular targets, or at least in a pharmaceutic session, you can figure out how to get antibodies inside cells in cell culture using by sort of protein transduction or even by sort of viral transduction. It's not so easy to do that in a targeted way in people, but maybe ultimately one would do that. And I think they do have some advantages over small molecules in terms of ease of being able to get high selectivity and maybe also to disrupt interactions where the interacting surfaces are relatively flat. Of course, they're not orally bioavailability. I think one significant limitation is in inefficient delivery across the blood-brain barrier. And colleagues at Genentech, as well as many other groups, including some in this room, have also been approaching this problem and have been able to increase the delivery of antibodies into the brain in animals by at least a few fold. I don't think we know yet as a field whether that's going to be enough. Certainly, there's a real need here to develop more efficient sort of methods. There's a lot of unmet need in neurodegenerative disease, Alzheimer's, Parkinson's, and other chronic neurodegenerative diseases. So in a way, kind of listing this sort of limitations could be sort of viewed as opportunity space for all of us in what is a fairly crowded and fairly mature field at this point. Another aspect related to delivery is around inefficient tissue penetration, and then resistance, of course, is sometimes observed, but this may relate to target selection. Can you address that with antibodies? Perhaps if one is used in combinations or multi-specific antibodies. But I think I sort of view this slide in a way as well. This is kind of our sort of new frontiers for us to explore as a field. So why develop improved antibody drugs? And I think really there are several compelling reasons to do that. I think one is that firstly it's feasible to do that. I think if you look at almost every session in this meeting, there's some aspect of manipulating antibodies to improve their properties. I think another, perhaps probably the most compelling driver is to increase the benefit to patients. From a commercial standpoint, one has to continue to improve for long-term commercial success. In this era, human antibody generation is routine, arguably even commoditized, and there's increasing competition. And one aspect of that competition is biosimilars. This table is taken from a review from Nature Reviews Drugs Discovery, and actually it's just a few examples here, but the point that was being made is that for many of these pioneer antibody drugs have either come off patent in major markets or will soon do so, and that's not escaped people's attention, and there's a lot of effort which is being devoted to developing biosimilars. One of these would be Remicade, which was approved in EU, I think, last year, and very recently in the United States. In enhancing the performance of antibodies, broadly you can think of this in three different ways. One is enhancing existing properties. One can give antibodies new properties, or one can use them in combination. For the remainder of this talk, I'm going to focus on one area, and that's bispecific antibodies. I'm going to start with some broad introductory remarks about the field, and then I'm going to use the second half of the talk to comment on some of our own work over the last few years. So bispecific antibodies is an area of the antibody field that has been really 50 years in the making. So the first concept of bispecific antibodies was described by Alfred Nisanoff and colleagues in 1960. And what they did was to take IgG molecules, proteolyze them, and reduce the Fab'2 fragments and recombine them. And they had the great insight at that time that would be a way to make bispecific antibodies. Of course, at that time, that was sort of predates the birth of a biotech or really just the sort of modern era of protein drugs. And so it took many years for the technology to catch up. And the first approval of bispecific drugs only came in the last few years. So there's a lot of white space in this slide, and obviously there are many things which kind of contributed to the approval of these bispecific drugs, and I've just called out a few major milestones along the way. One of these was from the late Cesar Milstein in the so-called hybrid hybridomas to make bispecific IgGs in the early 1980s, and then the advent of the BITE formats in the mid-90s, which the BITE's being used for Blincyto. In the sort of the biology fronts, the concept of retargeting T cells with bispecifics came from Michael Bevan in about 1985. It took some really bold Japanese investigators in their 1990 to actually explore this clinically, and actually this was in malignant glioma, and introducing the bispecific and activated T cells directly in the brain. And I can sort of remember this quite distinctly because Genentech at the time was trying to get the leaders interested in HER2, CD3 sort of bispecific, and I think this was just at that time we didn't have really the appetite as a company to pursue it, and I think it really a major sort of watershed in the field came with this sort of work of Micromet and later with Amgen in developing the BiTEs and ultimately the approval of Blincyto. So if you look now at their bispecifics which are in clinical development or in preclinical development, and ask, well, what are they being used for? The most common application clinically is in retargeting effector cells and mainly T cells, but also NK cells. Another popular application is in blockade of two different receptors or ligands. As for emerging applications, well, if I could, to get all of these on this slide, actually would require microscopically small pictures. So I've just picked a few, and you really have to look around at the agenda for this week to realize actually there are many, many different applications. I've mentioned one already with delivering antibodies into the brain. Another one that we're interested in is activating receptors. One, I think, which will be a really nice paper from the group at MedImmune in bispecific, or as they call them, biparatopic antibody drug conjugates. And I think there's a speaker later this week. And there are certainly sort of many other applications. In this slide, for convenience, I've shown all the bispecifics as IgGs, but in reality, there's an astonishing variety of different formats, and I can broadly classify them into bispecific IgGs, appended IgGs, bispecific antibody fragments, fusion proteins, and antibody conjugates. And at the time we wrote this review, which was about this time last year, there were about 60 different formats that we could find in the literature, and actually a number more published since, and probably quite a few that are not yet published. And of these, about 16 at that time had reached clinical trials. So why so many formats? Well, I think one really has to choose a format for the particular application. So thinking in terms of the valency of each arm and the particular application, the desired pharmacokinetic properties, maybe there are constraints around geometry. So I think it will take a few years really to sort out for different settings what formats work best. But in my opinion, it certainly won't be one size fits all. A format that we have, and many others have spent a lot of time in, is the bispecific IgG format. It has the attractive features of potentially long half-life, optional effector functions, and no artificial linkers, which could potentially be immunogenic or a source of aggregation. It's sort of well known that if you express two antibodies in the same cell, that in addition to the desired bispecific, one can get up to 9 unwanted species, which reflects that heavy chains can homodimerize in addition to heterodimerize, and in many cases, light chains can pair promiscuously with non-cognate heavy chains. Now, almost 20 years ago, we came up with the so-called knobs and holes technology to facilitate the heterodimerization of heavy chains, which simplifies this problem down to 3 unwanted chain pairings. Much more recently, Charlie Eigenbrot and Mark Ulch at Genentech solved the structure for knobs into holes containing FC, so that it could actually compare our original modeling with the structure, and actually they're kind of remarkably similar. This is an open book view of the knobs into holes CH3, and as we had predicted, these mutations are fully sort of buried. And at the time we were not sure if there was going to be any sort of untowards effects of these mutations. Certainly we knew in terms of FcRn binding, protein A binding, Fc gamma receptor binding, they were inert, but that's not the same thing as when you kind of take knobs into holes bispecifics into patients, that they would necessarily be sort of benign. However, we did treat quite a lot of patients with onatuzumab, or also known as METMAB, and saw a very low incidence of antibodies, and really the antibodies we saw were to their variable domains rather than the FC. So to the best of our knowledge, there aren't any significant liabilities from the knobs and holes mutations. So a few years ago, colleagues Christoph Spies and Justin Speer at Genentech took this one step further and actually took separately expressed 2 different half antibodies, separately expressed in single cells, and assembled them in vitro into bispecific IgG. This actually works very well, and we have 3 bispecific IgGs which are in phase 1 using this approach. It does have some drawbacks, however. The throughput is low, it's resource intensive, and we were particularly interested in kind of high throughput methods so you could actually screen bispecific, so kind of library screening. And so what you'd really like to be able to do is figure out how to express bispecifics in a single cell. And over the last few years, there have actually been a number of kind of clever approaches to this. I think starting with their colleagues at PRED with a kind of cross-mab technology, and then more recently groups from Lilly, from Amgen, and most recently from MedImmune, sort of different approaches. Next few slides, I'm going to show you what we've done, and I'll try and point out some places where I think our approach has some particularly advantageous features. We basically took 2 IgGs, kind of used the knobs and holes for driving the heavy chain heterodimerization. We computationally redesigned the CL-CH1 interface using the Rosetta program of David Baker. As I'll show you, that gave some benefit, but on its own was not sufficient. And so we sequentially made mutations at each of the other domain interfaces. And I'll show you, in the interest of time, I'll show you some of the most promising versions which have mutations at all 4 interfaces. We picked HER2, CD3 as the antibodies for a couple of reasons. One is that, well, we've worked with these antibodies for a long time and we know a lot about them. A more important reason was that we wanted to make this problem as difficult as possible, because we thought that if we did that, then if we found a solution that worked, that it might work broadly for other antibodies. And specifically, we picked these antibodies because there is no light chain pairing preference. It really is random within the limits of our data. What we're looking at here is our open book view of the CL and CH1, showing the sort of beta strands in gray, and in the sticks, the residues at that interface. And we used a knobs and holes strategy, either 1 knob and hole or 2 knobs and holes. And we allowed 9 or 10 residues to vary in the computation, so knob being a kind of a large aromatic residue, and the hole being a small residue such as alanine, serine, threonine, or valine. And so Rosetta simulates the structures and calculates the binding energy. We developed a so-called contrast filter, and after For the mutant structures, we would calculate the energy of this and also both of the potential sort of mispairs, and then calculated the energy difference from the mutant and the closest mispair. This is quite a computationally intensive calculation, and at the time it took us about 10 weeks. And frankly, we didn't have enough sort of computer firepower, so eventually we rented part of the Amazon cloud. So if you're going to do this at home, I would, in this era, it's pretty inexpensive to do this by renting a cloud from one of these commercial sort of vendors, and you can do what kind of took us 10 weeks, literally overnight. So these are the scoring from Rosetta. So these are in energy units and the contrast filter. These are not kcals per mole, they're kind of arbitrary units. But you're looking for large negative numbers. So in terms of stability, so the most stable would be on the left here, and for contrast, the biggest contrast will be at the bottom. So you'd really like things in this quadrant sort of down here. What we ended up choosing was, well, we didn't actually really have a sense of just, well, how much stability was going to vary. So we took things, a lot of cases with high contrast scores. We looked at the entire dendrogram of all of these variants and picked examples from all of the major branches. They were subject to gene synthesis, and we evaluated them. So our initial assays were to co-transfect these 4 different chains to protein A purify the mixture, and then quantify the bispecific IgG content by a sandwich ELISA. And we've done the controls to show actually, well, it only signals when you have both specificities present. Before we did that, we looked at the expression of these mutant IgGs, and frankly, with so many changes at their domain-domain interface, we thought there was quite high risk that we may have just completely messed things up and that they would be very poor expressors. Happily, that wasn't the case. There actually, virtually all of them were similar to wild type, with a few which were more poorly expressed. So then we did our sandwich ELISA to orientate you here. So the blue is the parent, so that basically is random pairing. The green is our in vitro assembly from 2 half antibodies that I described earlier in my talk, and you can see for these Rosetta designs, they're somewhere in between. And yes, we did actually look at 180, and they were pretty much basically all within this fairly narrow band. So we picked one of these called YT65 and characterized it much more extensively. And so, it's clear that this wasn't sufficient on its own. So in collaboration with Christoph Spieth's lab, we introduced some charge pairs in the variable domains, and we picked residues which had been widely known, actually, really for 15 or 20 years in being potentially amenable to mutagenesis and directing the specific assembly of different V domains. We made charge pair mutations, so mutating these glutamines to either glue or lyse in either orientation. So in parallel with this, we were also kind of working on the mass spec sort of methodology, and it was sort of obvious that, well, the bispecific ELISA wasn't terribly convenient, and we'd sort of reached the kind of limit of what you could do with a bispecific ELISA, and we certainly didn't have the appetite to do that for every bispecific that came along. So, but mass spec seemed like the sort of the natural tool to approach this problem. At the time, we were using the quadrupole time of flight, or so-called QTOF sort of technology, and you can actually resolve the different species, the 4 sort of main chain pairings, or they'll point out that actually 2 have exactly the same mass, so the desired bispecific and the chain scrambled. But I think as you're going to appreciate, these peaks are not fully resolved. So if you start to kind of engineer interfaces, and hopefully these peaks get smaller and this peak gets bigger, you really don't have a prayer of being able to accurately quantify, because you basically don't have a sort of clear baseline resolution. And so we were using the version of the extended mass range version of the Orbitrap. And using that technology, it really was possible to get real kind of baseline resolution, and we kind of set that up in a way that we were able to look at hundreds of samples, and that actually was really key to being able to analyze different designs. So now here we're looking at the wild type, and you can see there are three sort of major peaks, and you realize that, well, you actually have another problem here. You have these two peaks, the bispecific IgG and the light chains scrambled, which have exactly the same mass. However, if you know you quantify these other two peaks, you can mathematically estimate, well, how much is the bispecific and how much is a mispaired, and so for the wild type, it literally was about 25%. For the Rosetta design on its own, it brought it up to 67. For the charge pairs, variable domain charge pairs on their own, it was about 80. And then if you put them together, we ended up with about sort of 90. So certainly a step in the right direction, but not quite as good as what one would like. And you can see, well, this has mutations out at 3 of the 4 domain-domain interactions. So an obvious thing to do was to make changes at the other one as well as the other CH1 interface. And so we put in a charge pair there. This is going to be called V3 in the next couple of slides. And so for the HER2 CD3 example, we went from roughly low 90s% of the bispecific to within the limits of our data sort of quantitative assembly of the bispecific. So this is just one example. Of course, what you want to do now is to see, well, how well does that work across different antibodies? And we've looked at a few others thus far. And so all the ones we looked at so far, which have given quantitative or close to quantitative yield of the bispecific. Certainly they work somewhat better than the sort of prototype version, which had mutations, V1, which had engineering at 3 of the 4 domain-domain interactions. One thing which we also did was to look closely at the wild type. And certainly if one looks at the literature, there are hints that there can be cognate heavy light chain pairing preferences. However, without sort of the mass spec or without a kind of a way to be able to accurately quantify that, you don't actually really know how significant that is. So using the kind of disambiguation method that I described, we were able to kind of estimate that for some of our antibodies, as much as 70% of the bispecific is formed without actually making any mutations. So in other words, in some cases, there's an inherent chain preference. Extending this now to other human isotypes, so we've looked at G2 and G4, and actually worked quite well for both of those. And also, not shown on this slide, but we've also it's also been successful with murine IgG2A. So next we did some structural biology and solved some structures of these engineered variants. How much? 5 minutes, okay. We're in the home straight. So we solved some structures and to fairly high resolution, and actually they overlay very closely with the parent that was solved many years earlier. One of the first things we did was to compare the Rosetta structure with its model, and we were surprised really how close the fit was. One rotamer we got wrong, but the rest were astonishingly close overlay. We also looked at their charge pairs, and I'm kind of curious what was going on at the interface. Really, did you have sort of a hydrogen bond network across the interface? And the answer is, well, yes in one case, not in the other. So in one direction, with the lysine mutation on the VL and their glutamate mutation on the VH, the data are consistent with the hydrogen bond network across the interface. In the other direction, apparently just hydrogen bonding residues within VL and ordered waters. For the two CL-CH1 interfaces, there are clearly hydrogen bonds, and that's a sort of network across CL-CH1. we're also worried about sort of messing up stability or affinity, and so we also looked at that fairly closely, and I'm showing you some of the data here, and all of the variants that we've looked at so far have similar affinity for HER2 and melting temperatures that are similar or just slightly lower. We looked at B-cell killing assays and see similar levels of killing for the single-cell bispecifics, different versions with their in vitro assembled, shown in green here. The PK is also similar in SCID mice. We haven't looked in cynos yet, but that's, experiments are planned in the context of another bispecific. And then final data slide is to look at stable cell line production. What I've shown you thus far has all been with transients, and we've also now, this was originally with V1 because that's the first version that we had, shown that the stable cell line production is at least as good as the transients, and we're in progress have stable cell lines with their versions which have mutations in all the interfaces. And then finally, let me show this summary slide. So we have a design which, for making bispecific IgGs, which works quite well in the context of several different antibodies we've tried. It works for about 5 or 6 different antibodies we've tried, multiple human and isotypes, and similar affinities and TM to the wild type. And as I showed you in the last slide, initial data with in making cell lines also looks favorable. Finally, it's a pleasure to acknowledge my Genentech colleagues, particularly Iwan Dien, a very talented former postdoc in my lab, great collaboration with Christoph Spies, and really terrific collaboration with the mass spec group and many other collaborators who are shown here. So thank you for your attention.